everyone. My name is Melissa Wang, and today my group members Vamsi, Vuppala, Liz Cummings, and I will be discussing our chemical engineering senior design project, the microfluidic analysis of citrated whole blood. So the overall goal of our project was to design a microfluidic device that is able to analyze the clotting ability of a sample of citrated whole blood under flow conditions. The product we designed is called the Microfluidic Multi-Surface Coagulation Assay, or the micro-MCA. So why is it important to study blood clotting? Blood clotting, or coagulation, is an extremely complex biological process that plays a role in a number of diseases and disorders. They can range from too much clotting, which is what happens during heart attacks and strokes, to not enough clotting, which occurs in diseases like hemophilia. Patients who have coagulation disorders or are taking drugs that affect their blood clotting often need special consideration when it comes to medical treatment. So when designing the micro-MCA, our goal is to create an accurate and efficient diagnostic test. The micro-MCA is a single-use, disposable device that is able to analyze 1 milliliter of citrated whole blood over 4 separate clotting surfaces with a runtime of 5 minutes. So who's going to use the micro-MCA? Currently, the U.S. market for blood testing totals just under $14 billion, with a projected annual growth rate of 8.4%. More specifically, we will be focusing on the markets for clinical applications and point-of-care testing for the micro-MCA. The clinical applications market include any clinical trials related to blood clotting and treatment of patients who are currently on anti-clotting drugs. The point-of-care testing is diagnostic testing that takes place at or near the site of patient care. And in that category, we chose to include treatment for patients who are suffering from heart attacks and strokes, as well as trauma care, which include emergency room visits for injury and motor vehicle accidents. The total from these two categories is 57 million patients per year, and this indicates that the market for our product is strong and growing. To give some context for what our device can do, here is a chart comparing the micro-MCA to other diagnostic devices on the market. As you can see, the micro-MCA is the only device that does not require an incubation period. This means that there's no wait time for when the doctor draws the blood to when the analysis process can begin, which means that the entire processing time is shorter. Also, the micro-MCA is able to analyze a single sample over 4 separate clotting surfaces, which for other devices would require multiple tests. Finally, you can see that the micro-MCA is the only device that is capable of analyzing blood under flow conditions as well as simulate both clotting pathways that occur inside the human body, meaning that the results will be more biologically relevant. Here you can see a 3D model of the micro-MCA. On the left is the top view, and on the right is an isometric view, with the sides transparent so you can see the internal parts of our device. The micro-MCA is made up of a microfluidic chip that is mounted on top of a plastic cartridge. Microfluidics is the manipulation and control of fluids at a sub-milliliter scale, and the idea is to use this technology to create these small-scale devices in order to replace what are traditionally off-site and slow tests. Our microfluidic chip is made up of a layer of polydimethylsiloxane that has been patterned with the microfluidic channels. Underneath is a glass slide that has been layered with the 4 clotting surfaces. Vamsi will now be passing around some of the devices that we used during our lab test. The plastic cartridge for the micro-MCA is able to hold 4 pipette tips and has 3 wells. 2 of the wells will contain calcium buffer and fluorescent dye, the 2 reagents that are needed in the analysis process. You can see here a picture of some of the microfluidic devices that we used during lab. The 3rd well is where the doctor will place the blood sample, and after the blood sample has been deposited, the doctor will place the entire device into a processing unit, and the automated analysis process can occur. Liz will now go over the blood chemistry of clotting and how we modeled it inside of our device.
Thanks, Melissa. All right, so since the micro-MCA tests a sample of citrated whole blood, it's important to understand some of the basic chemistry of blood clotting. There are 2 pathways to blood clotting, which are the intrinsic pathway and the extrinsic pathway. The intrinsic pathway is activated by exposure of blood to a charged surface or collagen, while the extrinsic pathway is activated by the exposure of blood to tissue factor, which is what happens when you get a cut on your skin. After activation, a series of reactions occur which lead to the accumulation of thrombin. Thrombin then activates platelets and fibrin, which are 2 of the most important components of a blood clot. Calcium is required for most of these steps to occur, and since clotting is not desired for most medical testing, it's standard procedure in hospitals and clinics to add trisodium citrate to the blood as it's drawn from the body. This process, known as calcium chelation, basically binds up all the free calcium ions in the blood, which prevents clotting from occurring until calcium can be reintroduced into the blood. Since the micro-MCA tests coagulation, we want to introduce those calcium ions back into the blood, a process called recalcification. We're going to do this on chip, which not only simplifies the job of the doctor or physician, but eliminates the incubation time associated with blood testing. We are going to recalcify our blood by sheathing it on either side with a calcium buffer and enabling those small mobile calcium ions to diffuse back into the blood. We will then flow our recalcified blood over 3 clot-inducing surfaces. 2 of those surfaces have already been developed, which are the collagen surface and the collagen tissue factor surface. The 3rd surface we developed to test the intrinsic pathway on a medical device, which has not been done before. That's our collagen kaolin surface. Finally, we will analyze our blood using fluorescent intensity of platelets and fibrin as they accumulate on the clotting surfaces. Okay, so like I mentioned, we need to recalcify our blood on chip. We first modeled this diffusion of calcium using COMSOL and analytical methods to determine baseline parameters such as the angle of intersection and the length of time for which calcium needed to be in contact with the blood to reach adequate levels of calcium in the blood. Then we tested our designs in the lab. We sheathed our blood in a fluorescent dye and imaged the channel along its length. We converted this fluorescent diffusion data with the diffusivity of calcium and found that calcium should reach an adequate level in the blood within 2 centimeters of flow on chip. Finally, we tested our design with a calcium buffer and citrated whole blood. We placed our clotting surface 2 centimeters downstream, and we observed the accumulation of platelets and fibrin. This indicated that our design for on-chip recalcification was a success. Next step on our chip is the flow of those blood over those clot-inducing surfaces. And like I mentioned, we developed a novel kaolin, collagen, and lipid surface. This surface increased the accumulation of fibrin by a factor of 20 at an early time point of 5 minutes. In the context of the micro-MCA, this amounts to a very strong diagnostic signal at a very early time point compared to other devices currently on the market. Vamsi will now walk us through the chip.
Thank you, Liz. So let's take a look at the unit operations that will occur on the micro-MCA. So first, this is an image of what the micro-MCA cartridge device will look like right out of the packaging. There's green for this is the calcium buffer. This is the dye. We have pipette tips, and these banded surfaces are the clotting surfaces. So now, the first thing the doctor needs to do is add 1 ml of blood to the device and put it into the processing unit. The processing unit takes care of it from there. It uses a liquid handler arm to pick up dye and move it to the blood and mix it. From there, it moves that dye to the reservoir here on the microfluidic portion. And then it switches pipette heads, takes the calcium buffer, and adds it to the calcium buffer reservoirs. Now, once all the reservoirs are filled, the syringe pump begins to pull, causing flow down the microfluidic channels. And as the fluid flows, you're going to have recalcification occurring right here. And as recalcified blood passes over the surfaces, you'll have coagulation occurring. That's where the processing unit will image on our device. After the 5 minutes of runtime, the device is done. The doctor can dispose of the device in a biohazard bin, and the processing unit will read the images and looking at the fluorescent data, offer a diagnosis for the patients and how well the patient's blood coagulates. So, this here is a video of those images. This is blood flowing down the clotting surface. This clotting surface is the kaolin collagen clotting surface. The green is platelets that's coagulating there, and the red is fibrin forming a network on top of the platelets. The blood is flowing at a shear rate of 200 per second. This is the shear rate of blood flowing through your veins right now. So like I had mentioned before, the processing unit images these, and taking the fluorescent intensity data from these images, it's able to, it'll be able to compare blood that doesn't coagulate well to blood that does coagulate properly and offer a diagnosis. So far we showed the technical viability of our project. Our project also has a lot of financial viability. Our project, we designed the manufacturing to produce 1 million devices per year. We expect to sell the devices at between $30 and $100. This means we'll be getting profits annually of between $9 and $79 million. So what does this mean over the project lifetime? We expect a 10-year project lifetime, and we expect to see very strong internal rates of return and return on investment. So what have we shown you today? overall. We've demonstrated 2 brand new microfluidic processes. First being recalcification through diffusion in sheath flow. Second is the brand new clotting surface, K-Legend, lipid, and Kale. This kind of surface is not on the market right now. Taking these 2, we showed how we can make this a profitable venture. Overall, the micro-MCA can be used in the pharmaceutical industry to create the next warfarin-like drug for patients who are victims of heart attack and stroke. It'll help pharmaceutical companies create the next big medicine that will help them. It can also help doctors in trauma care units. When a patient comes into the ER and is profusely bleeding, they'll be able to test the patient and see how well their blood clots and perform a diagnosis and see how to treat the patient better, and they'll be able to do this faster. Overall, the micro-MCA is a fast, accurate, and economically feasible device that satisfies an unmet need in the market. We'd like to thank Dr. Diamond, who is our advisor, and he offered us this project. We'd also like to thank Dr. Cedar and Professor Fabiano for their advice and their mentorship throughout the semester as we were working on our project. Thank you, and we have some time for questions.
So what we saw, that's a one-time use?
It's a one-time use.
The ROI looks pretty impressive. Does that include, isn't there a bunch of additional startup costs in terms of FDA?
We do include all of that. We calculated a $3.5 million clinical and FDA cost that we did include. We also included the costs of equipment, manufacturer, like labor for the manufacturer and the labor for the sales force, as well as the cost for sites and retrofitting and all the taxes and inflation involved with pricing over the 10-year lifetime.
How hard would it be for Siemens or those other companies to just modify what they're doing today and do this?
So they could, but their devices currently measure one surface at a time, so they would have to change their entire product. They'd also have to change how the product was imaged in the machine itself. Yeah, so everybody would have to buy a new machine, and